Chapter 13, Section 2, The Structure of DNA. Learning objectives: identify the chemical compounds of DNA, describe the clues that help scientists determine the structure of DNA, and explain what the double helix model shows about DNA. DNA is an example of a nucleic acid, which is one of the four main categories of biological macromolecules. DNA is made up of nucleotides joined into long strands or chains by covalent bonds. Nucleic acids are long, slightly acidic molecules built from small monomers or subunits called nucleotides. Each DNA nucleotide is made up of three components: a 5-carbon sugar called deoxyribose, a phosphate group, and a nitrogenous base. Nucleotides join together to form strands of DNA. DNA uses four kinds of nitrogenous bases: adenine, guanine, cytosine, and thymine. Biologists often refer to the nucleotides in DNA by the first letters of the base names A, G, C, and T. The nucleotides are joined by covalent bonds between the sugar of one nucleotide and the phosphate group of the next. The nucleotides can join together in any order, meaning that any sequence of bases is possible. The four different nucleotides, like the 26 letters of the alphabet, could be strung together in many different sequences, so it was possible they could carry coded genetic information. One of the puzzling facts about DNA was a curious relationship between its nucleotides. Years earlier, biochemist Erwin Chargaff had discovered that the percentages of the bases adenine and thymine are almost equal in any sample of DNA. The same relationship holds for the other two nucleotides of guanine and cytosine. The observation that A equals T and G equals C became known as Chargaff's rule. Scientists showed that the DNA samples from organisms as different as bacteria and humans obeyed this rule. However, neither Chargaff nor anyone else at the time had the faintest idea of why. In the early 1950s, a British scientist named Rosalind Franklin began to study DNA. Franklin used a technique called X-ray diffraction to study the structure of DNA, the DNA molecule. First, she stretched DNA fibers into a thin glass tube so that most of the strands were parallel. Next, she aimed a powerful X-ray beam at the concentrated DNA samples and recorded the scattering pattern of X-rays on the film. Franklin worked hard to obtain better and better patterns from the DNA until the patterns became clear. In May 1952, she took an X-ray photograph known as Photo 51. By itself, Franklin's X-ray pattern does not reveal every detail of the structure of DNA. However, it does provide some very important clues. The X-shaped pattern shows that the strands of DNA are twisted around each other like the coils of a spring. This shape is known as the helix. Also, the dark spots at the top and the bottom show that the nitrogenous bases are stacked at regular intervals near the center of the molecule. While Franklin was continuing her research, American biologists named James Watson and British physicist Francis Crick were also trying to understand the structure of DNA. They built three-dimensional models of the molecule that were made of cardboard and wire. They twisted and stretched the models in various ways, but to their best efforts did nothing to explain the DNA's properties. Then, early in 1953, Watson was shown a copy of Franklin's remarkable double X-ray pattern. The effect was immediate. In his book, The Double Helix, Watson wrote, "The instant I saw the picture, my mouth fell open and my pulse began to race." Photo 51 contained the clues they needed to construct a model of DNA. The data in Franklin's X-ray pattern enabled Watson and Crick to build a model that explained the specific structures and properties of DNA. The pair published their results in a historic one-page paper in April of 1953. Watson and Crick's breakthrough model of the DNA was the double helix. The double helix is where two strands of nucleotide sequences are wound around each other. The double helix explains many of DNA's most important properties. The double helix model explains Chargaff's rule of base pairing and how the two strands of DNA are held together. The double helix model even hinted to how DNA can function as a carrier of genetic information. One of the most important aspects of the double helix model is that its two strands of DNA run in opposite directions. Evidence for this had appeared in an early X-ray pattern of DNA, which Franklin had taken almost a year before Photo 51. At first, Watson and Crick could not explain what forces held the two strands of DNA's double helix together. Then they discovered that hydrogen bonds could form between certain nitrogenous bases, providing just enough force to hold the two strands together. Recall that hydrogen bonds are relatively weak chemical forces. However, the hydrogen bonds form only between certain bases, such as adenine with thymine and guanine with cytosine. This nearly perfect fit between A, T, and G and C nucleotide is known as base pairing. Watson and Crick realized that base pairing explained Chargaff's rule. It gave a reason why A equals T and G equals C. For every adenine in a double-stranded DNA molecule, there has to be exactly one thymine. For each cytosine, there is one guanine. The ability of their model to explain Chargaff's observations increased Watson and Crick's confidence that they had come to the right conclusion, with the help of Rosalind Franklin. In 1962, the Nobel Prize was awarded to James Watson and Francis Crick, and to Franklin's associate Maurice Wilkins. Sadly, Franklin had died of cancer in 1958 and was not able to share the prize or fully appreciate the acclaim her work had earned.